Hello, I'm Ann Montgomery, Editor-in-Chief of BioProcess International Magazine. I'm here in Oakland, California this week at the BPI West Conference and Exposition. A number of speakers have agreed to share their insights with us. I hope you enjoy their comments and find them valuable as you approach your work.
Hi, I'm Sigma Mostafa. I'm Vice President for Upstream and Downstream Process Development at KBI Biopharma. We are a CDMO. We have 2 locations in North Carolina as well as in Colorado. Nice to be here and talking to you. The general definition of robustness obviously is ability of a process to tolerate the expected variabilities that comes from raw materials or process conditions or human factors or equipment. And essentially robustness is something we need to demonstrate prior to a molecule going into market. And a lot of the process characterization work that gets done later in the life cycle of a molecule ensures that data is generated. So what we are talking about here is bringing in the sort of studies of robustness as well as understanding around a cell line and a process very early on so that there aren't any surprises going at the late phase of the molecule. In general, with the QBD approach, there is that idea of building quality into the process. So from the very early stage of from pre-clinical trials to early stage clinical trials, we are definitely trying to make sure that the design of the studies that are being done are through proper statistical design so that we do collect data early through late phase to build in robustness again throughout the life cycle. Some of the things that help in that regard are some of the high throughput technologies, which allows us to look at a lot of different parameters, both in terms of the process as well as the analytics part, because there are high throughput technologies now in the analytical side, which allows us to basically get a lot of data, not compromise on the data amount as we are trying to move molecule faster through the early phase of development in phase 1 or so, because oftentimes the other approach that has been done is platform development. What that means is you have built a platform for upstream and downstream development, and you essentially put all your molecules through that particular platform, but as a result you get very limited information. You do not know how your cell line or your particular product react to different process parameter conditions. So by using high throughput technologies such as, for example, AMBER 250 milliliter bioreactors, microbioreactors can be used for some process characterization work as well as early stage development, which allows for essentially gaining a lot more information and not lose out on that information in the effort to move fast. So cell lines, obviously, in general, the technologies around, I think, vector design as well as the host designs have come a long way, and that has allowed us to achieve these very high titers that we now see. And often the discussion is, okay, now that upstream has achieved this 10 gram per liter, at least in fed batch, and maybe 30 gram per liter when you're talking about high cell density processes, that really the challenge is now on the downstream. However, having said that, the focus has really still been on productivity and not so much in terms of getting some other independent parameters that you can look at early on for a cell line to know whether it's going to be a robust cell line and be able to withstand the process variabilities that you can expect as you go to larger scales or you go to different sites, etc. So I think more understanding of let's say CHO cell lines and being able to characterize them more certainly helps, and there's certainly been a lot of proteomics related work being done, but still I don't think the understanding of product quality in terms of how specific cell lines or clones affect product quality, or this concept of cell line robustness have not been studied further. So I think there's still some improvement there that can be done. So we are, of course, a CDMO, so we work on 50-some molecules per year, and a lot of those molecules are actually where we are developing the cell lines in-house. The context here was that these were cell lines we had received from certain clients, and in choosing those final cell lines, the driver in most cases has been productivity, and not a lot of thought around how the cell line may perform under different process conditions and such. So, so our driver was essentially to understand more how we can early on sort of pick a cell line that will make the later phases of the work, including the characterization studies, easier. And we are not going to have a situation where the normal operating range and the proven acceptable range are very close to each other, because basically if you go slightly away from a certain pH range or slightly away from certain temperature range, due to your cell line characteristics, it will fail. So obviously for mammalian cell lines, still there is a sense of it's more of an art. We don't fully understand all the attributes of a cell line and what controls that. But again, building in some of that understanding is what we want to be able to do, and we're looking forward to collaborating with some groups as well, academic and industrial, to achieve that goal. I have conveyed sort of our learnings through all these molecules, and I think there's really more technologies that needs to come in. So I definitely encourage vendors who are looking into these areas of sort of inline, various parameter that gets tested or controlled nowadays, that we look at a wider range of factors to identify cell lines and processes that will inherently make the process more robust. Thank you.
Thank you.